Good morning, everybody, and welcome to the second day of the International αβT-cell B-cell depleted haploidentical transplantation symposium. This new technology has really transformed the way in which we perform haploidentical transplant. And certainly it has transformed the way in which we perform transplant at Stanford Children's Hospital, thanks to the outstanding work of Alicia Bertaina. And of course, we work on the shoulder of a giant, Franco Locatelli, who really opened the field of haploidentical stem cell transplantation. So the CDCM is incredibly proud to host this symposium, and more to come in the future, hopefully in person and hopefully in Italy. What is the CDCM? The CDCM is the Center for Definitive and Curative Medicine. I have the honor to be the director of the CDCM, as well as the co-director of the Stem Cell Institute. And the CDCM has been established when I came to Stanford to really facilitate and accelerate the translation from the bench to the bedside for cell and gene therapies. We rely on four major pillars: the people, the physician scientists who really have the drive and the dream to translate their discovery into drugs for patients with incurable diseases. We rely on the Laboratory for Cell and Gene Medicine, which does all the process development of our living drugs, as well as the manufacturing, the clinical trial program that is taking care of all the support for the phase 1 clinical trials. And of course, the fifth floor of the beautiful new Children's Hospital, which was built thanks to a donation from the David and Lucille Packard Children's Foundation to the Children's Hospital and to the CDCM specifically. So based on these four pillars, we have established a very rich pipeline of phase 1, first in human, first in children clinical trials. And you will hear some of them today. Some are already in the clinic. We have in the clinic 6 phase 1 clinical trials, investigator initiated. We have more than 10 industry sponsored. And this is thanks really to all the members of the CDCM that works very hard to bring this product to patients with very high unmet medical need. Among these phase 1 investigator-initiated trials, there are also 2 trials with T regulatory type 1 cells that are the cells that my team discovered many years ago, and I cannot be more proud that we have now these cells in the clinic, and you heard from Alicia the description of this product yesterday. We also had the opportunity to participate to 2 successful spin-offs. One was Jasper Therapeutic, that to which we transfer our IND for the antibody-based conditioning in SCID patients. And more recently, Graphite Bio, for which I'm a co-founder, that is on gene editing for hematological disorders. So the CDCM is really the home for every faculty and investigator at Stanford that wants to transfer their discovery to the bench side. And I think that this is a unique academic infrastructure very much needed for investigators that have this goal, because we know that industry is a fantastic partner for us, but sometimes these decisions by industry are driven by many factors which are not necessarily related to the need of the patients. So we think that to have an academic infrastructure is incredibly important and transformational, as we heard yesterday from many, many speakers. So without any further delay, I would like to thank you all for attending this international symposium and give the floor to Mike Pulsifer, who will chair the first section of this morning. Thank you so much. Thank you.
Yes, thank you so much, Maria Grazie. I kind of consider it the main event. We had a very similar session at TCCT a couple of years ago, but it was not quite the right session because it needed the donation that Brandon Triplett's going to be giving to us today. So it'll be a really outstanding session where we're going to try to answer the question that we all want to know. So we all love haplos. Haplo is kind of the new thing, and we're all quite enamored, and yet there are very different haplo approaches that have very different implications to the patient. And this outstanding session with world experts in each of these areas is going to have fun and try to approach this question. And we will start first with Franco Locatelli, who is professor of pediatrics at Sapienza University of Rome and director of the Department of Hem-Onc of the Jesu Bambino facility there. He will start and talk to us about αβT-cell CD19 depleted haplo transplantation. And then afterwards we'll do, Brandon and Ken will talk, and I'll introduce them briefly before each of their talks. We'll save questions to the very end. So with no further ado, Franco.
Many thanks for this nice introduction, and let me start thanking Alicia first of all, but all the other people of the Stanford University group, in particular Maria Grazia, for inviting me to this great symposium. And as just mentioned by Mike, I will address the issue of αβT-cell and B-cell depleted HLA haploidentical transplant in pediatric patients with either malignant or non-malignant disorders. And these are my disclosures that I leave for a few seconds for you. And now we can start from a basic concept, namely that there has been an evolution in the approaches of T-cell depletion over time, not to mention the old pioneer work done by many colleagues using the soybean lectin agglutination and resetting. In 1995, the transplant procedure after the T-cell depletion was mainly based on the selection of CD34 positive cells, this resulting into the infusion into the patients of the pure hematopoietic and stem cells and progenitor cells. But in 2010, thanks to our pioneering work, mainly done by Rupert and Grettinger in collaboration with people of Milteni, a new method of more selective T-cell depletion was developed, and it is based on the selective removal of the alpha-beta lymphocytes and of CD19 positive cells with the goal of preventing EBV-related lymphoproliferative disorders that can represent a significant complication in this particular transplant setting. And thanks to this approach, the patient can benefit from the infusion of not only hematopoietic stem cells and progenitor cells, but also from the adoptive transfer of major natural killer cells and gamma delta T lymphocytes that can contribute to the prevention and control of infection complication as well as also to the eradication of malignant cells escaping the preparative regimen. And indeed, if you look at this cartoon, you immediately realize that thanks to this approach, there is the possibility of infusing a high number of hematopoietic stem cells together with a high number of also gamma delta and NK elements. And more in particular, in our experience, the median number of gamma delta T lymphocytes and natural killer cells that we have been able to infuse are 8 and 32 million per kg of recipient body weight, with a great efficiency in terms of both alpha beta T-cell and B lymphocyte depletion. And working together with Rupert, we developed in Rome this approach that has been based on the use of a conditional regimen varying according to the regional disease of the patients, and on the use of pre-transplant serotherapy, namely anti-lymphocyte globulin at the dose of 4 milligrams per kg per day on day -4, -3, and -2. And after the infusion of the T-cell depleted graft, no post-transplant pharmacological GVHD prophylaxis is employed. I'd like to spend a few seconds to share with you the algorithm that we consider for the choice of the donor, given the priority of to the criteria of NK alloreactivity estimated according to the Kircher-Lagan model developed by Andrea Velarde and Perugia, given the priority to the KIR genotype B instead of the KIR genotype A, and also giving then the priority to a higher B content score. And the reason for doing this is related to the fact that we obtain evidence that donor with a KIR-B haplotype, and namely an haplotype containing more activating receptor, it is associated with a lower cumulative incidence of leukemia recurrence, resulting into a better probability of the event-free survival in children with acute lymphoblastic leukemia. So we initially reported the first 23 patients given this type of allograft for a non-malignant disorder in 2014, demonstrating that the probability of disease-free survival was in the order of 90%. And today I am glad to share with you an updated analysis of these children with non-malignant disorder. In particular, I will start from the subset of patients with Fanconi anemia that we published 1 year ago. In particular, we treated here in Rome 24 patients with Fanconi anemia, given exactly the T-cell depleted and B-cell depleted transplant that I was already commenting. And you can observe the clinical characteristics of these patients. 23 of them were transplanted because of severe bone marrow failure, while the remaining patients had a myelodysplastic syndrome occurring in the context of Fanconi anemia. And in all patients but 3, we used the combination of low-dose cyclophosphamide, 300 milligrams per meter square, fludarabine, and single-dose 200 centigrade total body irradiation. And you can observe that the graph composition is exactly in line with what I already commented a few seconds before. The cumulative incidence of acute GVHD any grade was 17%, and only one patient experienced chronic GVHD. And the probability of the event-free survival and the overall survival are 86 and 100% respectively. And in the estimation of the EFS probability, we consider that graft failure as an event, but it is important to notice that all the three patients who experience either primary or secondary graft failure were rescued by a second allograft, in 2 cases, again, an upload transplant from the other parent. More recently, we published the whole cohort of children with non-malignant disorder, of course, with the exception of the Fanconi anemia population. In particular, we reported on 70 patients, 38 of them affected by primary immune deficiency, 11 by red blood cell disorders, 16 by aplastic anemia, and 3 by metabolic disorders. And you can observe that the clinical characteristics of these patients, in particular, 21 of them had a severe combined immune deficiency, and 6 hemophagocytic lymphohistiocytosis. And you can observe that both the neutrophil and platelet recovery was extremely fast in these patients, with a tendency for a good recovery also in patients with a number of CD34 positive cell infused behind the first quartile. We observe a cumulative incidence of graft failure in the order of 30%, but it was mainly concentrated in children with either thalassemia or severe aplastic anemia or hemophagocytic lymphohistiocytosis. In the remaining children, the cumulative incidence was in the order of 14%. Again, also in this group of patients, the cumulative incidence of grade 2 to 4 acute GVHD is extremely low, in the order of 14%, and the probability of overall survival is confirmed to be a little bit higher than 90%. We recorded 4 transplant-related deaths due to disseminated adenovirus infection, CMV pneumonia, and 2 bacterial sepsis, and 1 case of PCP already present at time of the allograft. And you can observe that in the group of patients who didn't have any active or recent infection, the probability of overall survival approaches 95%, while the risk of transplant-related mortality is higher and very close to the statistical significant level in those patients who have a recently active infection at time of allograft. Again, the probability of disease-free survival is confirmed to be very high, 87%, while that of the EFS, considering the graft failure as an event, is in the order of 66%. Let me spend a few words also on the immune recovery that we investigated in these patients, because it's a general pattern for all children given this type of allograft. And you can observe that in the first 2 months after transplantation, the predominant lymphocyte subset in the T-cell compartment is represented by the gamma delta T lymphocytes, while if you look at the alpha beta T-cell recovery, you can observe that already at 6 months after transplantation, these patients had more than 1,000 alpha beta T lymphocytes with an even distribution between CD4 and CD8 cells. And also the recovery of B-cell was very good. We also evaluated the viral reactivation in these patients, and you can observe that the area under the curve of the adenovirus infection was higher than that of CMV, this reflecting the lack of a really effective pharmacological therapy for these patients. Some years ago, we also published the initial results on 80 children with acute leukemia. In particular, 70% had ALL and 30 AML. And in that study, we demonstrated a leukemia-free survival probability in the order of 70% with no difference between ALL and AML. And you can observe that the main cause of treatment failure was leukemia recurrence, the cumulative incidence of relapse approaching 25%. Again, I am glad to share with you the updated analysis now enrolling 150 patients, and I decided to show you the data only of those patients who have at least 1 year and a half of follow-up to have solid and robust data. 106 of these 150 patients had acute lymphoblastic leukemia, mainly of the B-cell differentiation lineage, and 26, 64, and 16 were transplanted in CR1, CR2, and CR3. Of course, children transplanted in CR1 had a very high level of minimal residual disease at the end of either induction or consolidation therapy, or had a very poor raised molecular abnormalities, providing the indication for transplantation in CR1. 44 children were transplanted for AML, 29 in CR1 and 15 in CR2, and many of these patients had a very poor risk cytogenetic characteristics. Again, the hematopoietic recovery was very fast, the median time to platelet and neutrophil recovery being 11 and 13 days, and also the cumulative incidence of GVHD confirms to be very low. Indeed, only 3 out of the 150 patients experienced grade 3 to 4 acute GVHD. Also, in this enlarged cohort of patients and with the longer follow-up, the cumulative incidence of non-relapse mortality remains very low. Only 6 out of the 150 patients died, and you can observe the causes of death in this box. And the cumulative incidence of leukemia recurrence is exactly the same that we reported in the paper we published in Blood. And the median time to recurrence is at 6 months after the allograft. And indeed, you can observe that after 2 years following the allograft, very few recurrences were reported. The probability of overall survival in these 150 patients is 67%, while the probability of the disease-free survival is 71%. We also calculated the GVHD relapse-free survival that approaches 66%. And you can observe that for patients transplanted in CR1 and CR2, the DFS probability is in the order of 80%. We also evaluated the role played by the conditional regimen, and we found an advantage for patients given TBI as part of the myeloablation. But this advantage is entirely due to the use of radiotherapy in children with acute lymphoblastic leukemia, while we didn't see any difference in terms of DFS according to the use of radiotherapy in children with AML. Together with Irma Iroldi and Lorenzo Moretti, Alice and I had the opportunity to work also on the reconstitution of gamma delta T lymphocytes, demonstrating not only that B delta 1 and B delta 2 T cells promptly reconstitute in these children, but also that there is a subsequent decline in the percentage of these cells, and in the subset of children experiencing CMV reactivation, there is a predominance of the B delta 1 subset, while in the remaining patients who didn't experience CMV reactivation, the predominant subset is represented by the B delta 2 lymphocytes. And we also demonstrated that the use of biphosphonate, and in particular of zoledronic acid, is able to sensitize both myeloid and lymphoid blast to the lytic activity displayed by the gamma delta T lymphocytes. So this biological observation provided the rationale for considering the use of zoledronic infusion after transplantation. And you can see that the use of the zoledronic acid is associated with a lower cumulative incidence of acute GVHD, as well as also with a better probability of surviving to transplantation. And in particular, in 46 patients that entered into a trial, and may include children transplanted in CR3 or in CR2, but with persistent MRD before transplantation, the use of at least 3 doses of zoledronic acid 1 month apart, starting at day +40 after transplantation, is associated with a better outcome. Considering that there is still room for improving the reconstitution of adaptive immunity, we developed a study in collaboration with the Belgian pharmaceutical company for infusing a titrated number of donor T-cell transfused with the suicide gene represented by the inducible Cas9, and in particular the infusion of the genetically modified T-cells was scheduled at 2 weeks after transplantation. And we demonstrated that in children with acute myeloid leukemia, the infusion of this titrated number of alpha beta donor T cells was associated with a better probability of overall survival in comparison to the historical cohort that we published in Blood 4 years ago. And the main driver of the donor T-cell expansion was represented by CMV reactivation. Indeed, you can observe that the orange curve depicts the expansion of these caspacyte cells in children reactivating CMV expansion. More recently, we completed also a study evaluating the SJ track and the beta track recovery, namely the thymic output in these patients, and has also observed in the experience of our colleagues at the San Raffaele Hospital in Milan, who work on the infusion of donor T lymphocytes modified with another suicide gene, namely the thymidine kinase. The infusion of these caspacyte cells was associated with a much better SJ track and beta track already at 1, 3, and 6 months after the infusion of the cells. This clearly indicating that this approach can contribute to accelerate the recovery of adaptive immunity in patients given this type of allograft. This said, I would stop here. I'm more than happy to take any question you have, but let me thank all the people who are now working on this program, in particular in the clinic unit, Pietro Merli, Mattia Algeri, Francesca Del Bufalo, Federica Galaverna, and Dario Pagliara. I'd like to acknowledge the great contribution given by Concetto Quintarelli, Biagio De Angelis, Enrico Velardi, who worked together with Maria Luigia Catanuso on the SJ track and beta track project that I just presented, and Valentina Bertaina, who monitor the patients after transplantation. And I'd like finally to acknowledge the fantastic work done by the graft manipulation group coordinated by Giusy Lipira and finally I'd like to thank Lorenzo Moretta and all the other people for the continuous support in the selection of the best donor to be employed. Thank you so much.
Franco, that was absolutely outstanding. Really appreciate it. Mature data, always fun to see an update. Again, I think we'll save questions to the end, although I have a big long list of them already, Franco. Why don't we go ahead and move on? We're going to hear next from Brandon Triplett, who is an associate member at St. Jude and chief of the bone marrow transplant clinical service. He is going to speak to us about the next variation in T-cell depletion with CD45RA depleted haploidentical transplantation. So Brandon, take it away.
Yeah, I'm going to talk about our initial experience with CD45RA depletion and our T-cell depleted haploidentical donor transplants. Here are my disclosures. It's essentially some registration travel support to present our data at EBMT. And of course, you're all here to hear about haploidentical donors. And this is just a reminder of why we find it so interesting. Of course, they're highly motivated for us pediatricians because they're usually their parents. They're immediately available and can give large cell doses and can give additional collections. As you all know, HLA mismatch essentially causes all the main problems in transplant that we try to avoid, except for it can increase graft versus leukemia effect and see a reduction in relapse. And so our challenge here today that we're discussing yesterday and today is how to mitigate all of these bad things that happen when we increase alloreactivity, but maintain as much graft versus leukemia effect as possible. And so I think it's always important to look back at where we've been to know where we are and where we're going. And we really developed our haploidentical transplant program, really developed with the arrival of Rupert Hangrettinger and took off. And as you can see here, we started really with kind of myeloablative TBI-based regimens and mostly CD34 depletion. And we moved to more chemotherapy-based sub-myeloablative doses of chemotherapy with CD3 depletion. We did try some conventional T-cell add-back, but then moved away from that. We also tried to intensify the reduced intensity conditioning regimen by adding clofarabine instead of fludarabine. It was tolerable, but really wasn't having a measurable effect on relapse rates. And then OK23 went off the market, so we tried other antibodies again. And what we learned from these iterative series of protocols was that, this is all feasible, and we know that here in this meeting. We liked the submyeloablative chemotherapy-based regimens because of the lower treatment-related mortality. However, with all of these regimens, infections, particularly viral infections, and relapse remain primary causes of failure. When we tried conventional T-cell add-back, it was limited by graft versus host disease. And so even at a very low dose of 0.1 million T-cells per kilogram or 10 to the 5th cells per kilogram, anything above that, and the vast majority of patients got some form of graft versus host disease, and the rate of severe acute and chronic graft versus host disease was very high. And so we knew that we were limited by how many T-cells we could add if we're adding conventional T-cells. And so the question becomes then, can we do a more selective form of T-cell depletion? And of course, over the last day, yesterday and the start of today, we've heard about a form of selective T-cell depletion. In fact, the title of the symposium includes a form of selective T-cell depletion that allows gamma delta T-cells to be in the graft, and that's great. And we decided to look at a little different way to do selective T-cell depletion and explore that here at St. Jude. And so when you look at CD45, it's expressed on nearly all nucleated blood cells, and it comes in different isoforms. And for our purposes, the CD45RA isoform and the RO isoform are what we're looking at, because RA is on naive cells. And we feel that naive cells, most agree that naive cells are the primary driver of graft versus host disease. And if you look at memory cells in the preclinical setting, we know that they're less alloreactive, so should cause less graft versus host disease. They obviously maintain their viral specific activity. I mean, that's their job, and all of us, is to fight viruses. And again, in preclinical setting, they can mediate graft versus leukemia effect. So we felt that this was a good population to look at. And so we developed a novel selective T-cell depleted haploid transplant platform for children with hematologic malignancies that utilize CD45RA depletion. So I'll go through our rationale for our regimen a little bit here. We were concerned about using any antibody-based treatment. We talked a lot about ATG yesterday, I know, which was great discussions, and it was very helpful. And here, it made no sense to give any type of antibody close to transplant, because we would potentially kill the memory cells that were going to be in the graft. And so we needed an alternative to induce tolerance and get our grafts in consistently. And so we chose to go with a total lymphoid irradiation and as a replacement for serotherapy. We also wanted to make sure that we had a kind of a standard progenitor cell graft, because although there are progenitor cells after CD45RA depletion, there is some loss. And it was unclear as we developed the study if we were going to lose important progenitor cells with that depletion, because some of them are CD45RA positive. And so we went with a CD34 enriched graft because we felt it scientifically it would be more interesting if all of the effector cells came from the CD45RA depleted graft. It would give us a better understanding of the contribution of that graft to the early immune reconstitution. Now there is some, so of course the next graft was CD45RA depleted. So this was GCSF mobilized peripheral blood, apheresis, leukapheresis collection, and then underwent CD45RA depletion and then infused fresh. There is loss of, there is some loss of B cells, and there is some loss of NK cells with CD45RA depletion. And of course, CD34 has none of those cells. And so we did give an additional NK cell graft early post-transplant. Our GVH prophylaxis was primarily MMF for a short course after infusion. This study was for first allo transplant only, and we had 2 arms. Our initial few patients, we were treating only refractory patients. We wanted to make sure that this was safe and feasible, and then expanded it to all kind of standard patients who needed a transplant but lacked a matched donor. So I'm a visual person, and I just want to show visually the difference between our previous technique, which was CD3 depletion. You can see here in gray, this represents all of the donor T cells that we collect from a donor. And the red dot there is how many T cells were left. And so again, so a median of about 50,000 T cells per kilo, so on the range of 10 to the 4. And you can see in our overall range, again, we cap that at 10 to the 5th T cells per kilo, or 0.1 million per kilo. With CD45 RA depletion in this study, we actually give tens of millions per kilo of T cells on day 0. And actually in some patients, they received hundreds of millions per kilo. So, 10 to the 7 to 10 to the 8 total T cells were infused. Because we infused such a high number of T cells, not surprisingly, we saw a robust T cell engraftment. You can see previously here in red with our CD3 depleted protocols, our patients had essentially no detectable T cells at day 30, whereas in our patients who received 45 RA depleted grafts, you can see a pretty impressive number of T cells present immediately after engraftment. And the number of T cells stayed high throughout the first 6 months prior to de novo T cell production. We also saw a more rapid B cell recovery. And that's primarily not because of the graft itself, but because we no longer needed to use rituximab as EBV prophylaxis in the trial, which suppressed B cell recovery in the CD3 depleted trials, which included rituximab. And you can see that both CD4 positive T cells and CD8 positive T cells had more robust early engraftment. So for CD4, the pattern was that it was higher immediately at day 30 and remained higher throughout. And for CD8, we see what may be some type of proliferation, whether it's antigen-induced, it's unclear, but definitely some early proliferation of CD8 positive T cells that kind of settle down through the first 6 months post-transplant. So I know this is a very busy slide, but I just want to give you a feel for what we're seeing here. So on the top line, these are T cell populations, and the top row are the T cells from the donor graft that we collected. This is a representative sample. And then on the second line below, this is the same donor graft after CD45 or RA depletion. So you can see effective depletion of T cells that express CD45 RA. But the most intriguing thing to me is you see that the T cells then collected from the same patient who received this graft at day 30 and day 60 is a direct recapitulation of the cells that were infused from the RA depleted graft. And you can see again, CD8 cells are relatively depleted in the graft, but then proliferate significantly by day 30. But the population of T cells that we see for the first 2 months after transplant are exactly the cells that we put in with the RA depleted graft. And so if you look at this on average, what type of cells are we seeing? You can see here that, marked in red are the naive cells, the CD45 RA positive, CCR7 positive as one marker for naive T cells. And you can see in both CD4 and CD8 fractions, the donor has, 30 to 40% naive T cells. And those are basically gone for the first few months after transplant. And the T cell populations are made up primarily of effector memory T cells and central memory T cells. Around day 180, we start to see some naive T cells again, and that corresponds with de novo T cell production. And so the adoptive transfer of these memory cells really provides those T cell counts throughout the first 6 months that we showed before. And again, these cells, the memory cells, their job in all of us is to remember, prior vaccination, obviously, but also prior viral infections and mount a response. And so we looked at viremia, and we saw a marked reduction in viremia. Here you can see CMV and adenovirus, where we saw big changes. And not only were there less patients afflicted by viremia, but the peak viral load was significantly less in patients. You can see on the right-hand columns on the patients who got CD4, CD45 already depleted grafts compared to patients in our historical cohorts that got CD3 depleted grafts. And so this was true for CMV and adenovirus. Not shown here, but in the paper, you can also see there was also a marked reduction in the duration of viremia. And so in our previous CD3 depleted cohorts, viremia often lasted 2 months or longer, particularly with CMV, and that was extremely rare in CD45 already depleted cohorts. And so we have both protection from viremia and also for the patients who got viremia, it was much lower peak and much shorter duration compared to our historic patients. And we didn't have this data available at the time of the publication, but since then we've developed ELISpot assays to measure direct viral specific cytotoxic T lymphocyte expansion in these patients. And you can see here on the left, for patients who are donor positive and recipient seropositive prior to transplant, you can see that there is an expansion of CMV specific CTLs as early as 1 month post-transplant. So several patients by the day 30 time point had already expanded CMV-specific CTLs. And so this is just within a couple of weeks of neutrophil engraftment, we're already seeing viral-specific expansion. And by 2 months, essentially every patient who ended up with viremia, here denoted in green, had significant expansion of viral-specific T cells. And so it's a very early and very robust antiviral response that we're seeing in this protocol. So with these improvements in viral control, it was important to look at, the initial clinical outcomes. And so once we had 50 patients that had enough clinical follow-up, we decided to take a look at them. You can see here on the left, again, this protocol included both standard patients and refractory patients. And so refractory patients are patients who were unable to achieve remission, despite everything that could be tried. In fact, our referral pattern is such that many of the patients who end up coming for transplant came to our leukemia service first to get a salvage protocol because they were refractory to salvage treatments at their home institution. And so many of our patients, even who are able to achieve remission, were actually still MRD positive, as you can see here. 10 out of the 36 patients in remission had detectable disease, but had exhausted all kind of promising trials or therapies to achieve MRD negative CR. Our goal is definitely MRD negative CR, of course. If you look at our overall survival and event-free survival, of course, the thing that stands out is it's dramatically different between patients who are able to achieve CR and those who are refractory. And that's not surprising. We know that refractory patients are very difficult to cure. And we also saw that, we felt like this was better outcomes for our standard patients than we had seen previously. So we were very happy with these results and wanted to compare these results to our previous patients. So we look back, again, I described the evolution of T-cell depleted haplotransplants at St. Jude over the last 15 years. And here you can see we broke patients up into two groups. The blue group here, the blue dotted line we're describing as our initial haplotransplant. Those are patients transplanted basically 2001 to 2004 or 5. Those were our initial cohorts, mainly TBI-based regimens, mainly CD34 enrichment versus CD3. And then in red, these red lines you can see are the next group of patients. So this is essentially the 7 or so years before the current trial. And they got CD3 depletion. But these patients also received all of the same supportive care. And so weekly viral monitoring, the same antimicrobial prophylaxis, all the supportive care. We had all the antifungals and antivirals and things like that you would need. And so very similar supportive care. The main difference would be the regimen. when you look at graft versus host disease, you can see that there wasn't a statistically significant difference here in patients who received the CD45RA depleted transplant versus our prior transplants. When you look at transplant-related mortality and relapse, we looked at only patients in remission for this analysis. Patients who are refractory, first of all, we had small numbers, but we also know that patients who are refractory have both higher risk of mortality and higher risk of relapse. So looking at just the patients who are in remission, you can see a significant reduction in the rate of transplant-related mortality in this population and a trend towards lower relapse in these patients. And since we have lower TRM and lower relapse, you can see that our overall survival is much improved. So again, just as a reminder, this whole population of patients is just right at 15, 16 years. So it's a relatively short amount of time when it comes to talking about the type of supportive care that we have available. And so you can see both overall survival, and in this case, event-free survival is alive without relapse, which is our goal. We want to cure our patients. And so you can see a clinically significant and statistically significant improvement in both overall survival and survival without relapse in these patients compared to our prior protocols. We also looked at a contemporary HLA matched donor cohort. Of course, it's not perfectly the same type of patients. Most of our HLA matched donor transplants are patients who are local, and there's less of the high risk patients, but it's hard to normalize that. So we took all patients who were in remission and received matched donor transplant during the exact same time frame as patients in remission received haploidentical donor transplant on this protocol. And you can see outcomes are very similar. Both groups have a very low rate of TRM. And you can see might be a trend towards slightly lower relapse in the haplo cohort, but it did not reach significance. And so that's something we'll continue to follow. So this study, the accrual is complete. We have completed after 72 evaluable patients. Again, just to point out, very high risk population, although 53 of the 72 were in remission, over 20 of them had detectable disease at the time of transplant despite our best efforts. And so over half the patients actually, when you combine MRD positive and the refractory patients had detectable disease at time of transplant. And as a teaser, we'll have a clinical outcomes update presented at ASH by Dr. Swati Naik. So what have we learned from this initial study? I mentioned before the characteristics of memory cells are that they're less alloreactive, they obviously maintain viral specific activity, and can mediate graft versus leukemia effects. So what did we find? Well, we found that we can give more T cells without causing excessive graft versus host disease. Now, of course, excessive is in the eye of the beholder. What is excessive graft versus host disease really, depends on your goals. And depends on how treatable the graft versus host disease is. So, we could certainly argue about what is too much or not enough graft versus host disease in these very high-risk patients. We'll talk a little bit more about that in another slide. We were able to achieve consistent engraftment. We had a much more robust polyclonal T-cell reconstitution. I didn't show the V-beta spectrotyping, but we have a very diverse polyclonal T-cell population that occurs prior to any signs of de novo T-cell production. And so we have shown that in our publications. A marked reduction in viral infections. And there's a signal that there might be some GVL here from these cells with a possible reduction in relapse. That's something that we'll have to continue to follow. So I know the title of this session is really what's better, TCR alpha beta depletion or CD45RA depletion. So my bias is that, why not both? I think that combination therapy is the future. When we transplanters learn from our mentors who helped develop combination chemotherapy, for ALL, for example, or for lymphomas, the tenets of combination chemotherapy is non-cross resistance and non-overlapping toxicity. And so non-cross resistance, we know we have to attack these cancers from different ways with different mechanisms, because as if anything, CAR T-cell has taught us is that these cancers are extremely malleable, and they have a strong survival signal, and they can mutate. And so we have to attack them from more than one direction. That's an absolute. And we can't have overlapping toxicities. And so obviously T cells have toxicities. All of our cell therapies have toxicities, and they can't compound each other. We have to balance those out. And so I think combination chemotherapy with the right combinations is where we should go. And so what we're studying now is combining TCR alpha beta depletion, which allows us to give gamma delta T cells, as you guys have heard, with CD45RA negative depletion, which allows us to give memory cells, as I've just shown. In our current study, we're also giving a BiTE, blinatumomab, for patients who have CD19 positive disease. So if you look at the blinatumomab literature, you see that the main effector cells that utilize blinatumomab for tumor killing are effector memory cells. And I just showed you we have large numbers of effector memory cells in the early post-transplant period. So it made a strong rationale for utilizing a prophylactic BiTE therapy in the early post-transplant period. And so we really decided to study this combination going forward. And we also felt it was important to study it in different ways. And so we really are taking two pathways, if you will, for studying this combination of therapy. The HAP pathway is for standard patients who are in remission. As I showed, we were able to get very good outcomes without the use of total body myeloablative TBI. So the question is, can we maintain that very good outcome and really push to the lowest possible rate of TRM? So if we want to keep our low TRM in these patients, we have to continue to have enhanced reconstitution and especially viral control. So we have to maintain that piece of it. But can we reduce the morbidity and the small amount of mortality that we're seeing with graft-versus-host disease? So we would like to see less graft-versus-host disease if possible, both acute and chronic. And also we saw a lot of cytokine release syndrome and Graft syndrome, again, from giving these very large doses of T cells. And so we have an ongoing study called HAP2, where we're really trying to look at ways of combining, again, these three: alpha beta depletion, CD45RA depletion, and blinatumomab when you have CD19 disease, and try to accomplish these goals. On the other side, as you saw, our patients who have refractory disease still do extremely poorly, despite our best efforts. And we really must improve those outcomes. And this is a population I feel like we can really study new ideas and push for the absolute maximal graft versus leukemia effect. Obviously, we must keep the TRM low. If a patient dies from our treatment, then we obviously have not saved them, we have not cured them. And so it has to be kept low. Some graft versus host disease is acceptable as long as it's mostly treatable and reversible. And I didn't show that data, but actually even the patients on our current trial who got severe grade 3 to 4 graft versus host disease, most of them actually recovered, which was intriguing and something that we'll have to follow and make sure that holds up. But as long as it's treatable and reversible, and if having a little bit higher graft versus host disease rate adds to graft versus leukemia effect, again, it may be acceptable. We have to figure that out as we treat the patients. What is enough or too much graft versus host disease? And so we developed a strategy for those patients as well. I can show you a little bit about that. So back to the HAP patients, these are the ones with who are in remission at the time of transplant. And so they got our very similar reduced intensity conditioning regimen. They did get distal low dose ATG because we wanted to avoid radiation. In this case, we're trying to reduce morbidity as much as possible. They got a TCR alpha beta depleted progenitor cell graft. And then post-engraftment, CD45RA depleted DLI. And so we got consistent engraftment, and all patients in these 30 patients were able to receive their DLI per protocol. And you can see the doses here. And so you heard a lot yesterday about CD45RA depleted DLI. And we started at a similar dose at that 0.1 million per kilo. So this is the 10 to the 5th per kilo dose. And we knew that was, again, the threshold that was safe and conventional T cells. So it obviously should be safe with memory T cells. And then after 10 patients moved to dose level 2, which is 10 to the 6th or 1 million per kilo. And then after 10 patients moved to dose level 3, which was 10 to the 7 or 10 million per kilo. So really push the dose here. And we were comfortable doing that based on our experience with the prior study that I discussed. Now we have seen a handful of severe graft versus host disease. So we'll have to monitor that. Interestingly, it was not DLI dose dependent. So we have not seen a threshold in all of our studies, and we've given a lot of memory cell DLI in this and other studies. We have not identified a threshold dose of memory cell DLI that causes graft versus host disease, like you can define with conventional T cells. So it's really all I can say about this. We have some exciting data that's emerging. So hopefully you'll stop by our poster at ASH to look at that data. And the main data that we'll see is that there was a significant elevation in T cells after DLI. So the DLI here at the doses we're giving both increase the number of T cells and the number of CTLs. So we have some time here. I want to go through a single patient example as a proof of principle. So I think this was interesting. So this is a patient treated on our HAP trial. This is ELISpot data. So this is detection of expanded viral-specific cytotoxic T lymphocytes. And you can see here from the donor has detectable CTLs against various viruses, including COVID-19. So the donor did not provide us a history of having been sick with COVID-19. And this was prior to vaccination campaigns kicking in. So it appears that the donor did get exposed to COVID-19 because they had anti-COVID-19 T cells. So the patient underwent transplant. At 1 month post-transplant, there was essentially no T cell activity. All of the T cells at this point were basically gamma delta T cells. The patient received the DLI, the memory DLI, at that time point. And then 1 month after DLI, 2 months post-transplant, we see some activity against BK virus that actually got stronger over the next month. And then at 3 months, 2 months after DLI, 3 months after transplant, we see significant jump in activity against adenovirus. And both the adenovirus and BK virus specific CTL populations decreased by 6 months. And so it appears that the immune system had a memory response to BK and adeno. Interestingly, this patient never had any symptoms, never had identified viremia. We don't test for BK virus unless you have hemorrhagic cystitis, so we don't know what the viral loads were, but he never had any symptoms and never had any hemorrhagic cystitis. For adenovirus, we do test weekly for the first 100 days for adenovirus. He never had detectable adenovirus, never had enteritis or any of those symptoms. And so you can see here, these memory cells are basically, kicking in to these viruses and clearing them without us ever even knowing that they were there. And also interestingly, this patient did get a COVID vaccination at 4 and 5 months post-transplant. So before any sign of de novo T cell production, we are vaccinating our patients early, even though we know that responses may be suboptimal in this early period. And yet you can see here a stimulation of these pre-existing COVID-19 specific CTLs. So just a proof of principle of how the DLI can work in our patients. Finally, in the last few minutes, just show a quick brief preliminary data on our REF protocol. So again, in this case, what we're looking at here is patients who had relapsed after prior transplant. So on the left panel, you can see we looked at our data at patients who received first transplant at St. Jude and went on to relapse. What happened to those patients from the point of relapse? And you can see here on the left, the outcomes are very poor. This is from 1990 through 2018, I think. And this is 220 patients who relapsed after prior transplant. So patients who were able to receive a second transplant did better than those who were unable to. But even those who received second transplant, you can see, overall survival was below 30%. And so when you look at patients who went to our REF trial because of relapse or being refractory after prior transplant, We're seeing maybe perhaps a little bit better overall survival. This is preliminary data, but we're holding at around 50% overall survival. So perhaps a little bit better. I can tell you that non-relapse mortality has been actually pretty good in this very heavily pretreated population, and relapse remains a primary cause of failure. So we still have work to do in this population. And that's, really what we're here for on this symposium. I am extremely honored to be a part of this symposium. When I saw the lineup, it's just an amazing lineup of speakers. I've gotten a lot out of every speaker so far yesterday, and I'm really looking forward to the rest of today. I believe strongly that this is a platform. This platform that we're talking about is a great platform for adding additional immune therapies, whether they're cellular therapies that we isolate, as we've discussed, whether it's cellular therapies that we can manufacture now and in the near future, or non-cellular therapies, as we've discussed. I think there is just so much potential in this platform, and I'm very excited that we have this symposium. We have all you here watching this symposium, hopefully coming up with great ideas and doing your own studies. We can all learn from each other and really advance transplant. So with that, I'm almost out of time here, but obviously you can't do all this work without an amazing team. We have a huge team, both within our department and outside our department, many research collaborators to help us learn more about how this all works. Of course, we couldn't do it without our patients and families. Thank you.
Thank you, Brandon. That was outstanding. A lot of really great insight into very challenging patients with cancer. So our final talk in this fabulous session will be from Ken Cooke. Ken I know very well. We trained both in residency and fellowship together. Ken comes to us as a professor and director of pediatric bone marrow transplantation at Hopkins, and he will be talking to us about haploidentical transplants with post-transplant cyclophosphamide.
So Mike, thank you again. Really delighted to be here today. And I wanted to thank Alice and all of the organizers for inviting me to be a part of this symposium, in particular, being a part of this session. So I have the really distinct honor on behalf of my colleagues here at Hopkins and really many collaborators to talk to you all about haploidentical transplant with post-transplant cytoxan, which we clearly feel has been changing the standard of care. I really have nothing to disclose for this presentation, except, as Mike knows, I really like to talk, especially about things I'm passionate about, and it happens to be my birthday today. So Mike, later, be gentle on the questions, okay? So here are some objectives today. Want to describe in brief the process of allogeneic transplant using post-transplant cytoxan as part of GVHD prophylaxis. We'll define in a couple of slides the mechanism by which we think PT-Cy exerts its immune modulatory effects. Now, I'm going to identify the global impact of PT-Cy, and this is going to be a 30,000-foot look on some things. The literature, the body of literature is just overwhelming. We'll talk a little bit about that, but I'll summarize some things that I really think have been driven home by several investigators over the last decade or so, and then also illustrate advances made to the stem cell transplant procedure here at Hopkins and really plug some of my colleagues in the terrific work that they are doing. So, by means of introduction, we all know that allogeneic transplant is a important curative therapy for many pediatric and adult patients with malignant and non-malignant disorders. And historically, we've gone with the no guts, no glory, superlethal conditioning, HLA-matched donors. But we know that the use of reduced intensity conditioning and alternative donors have really improved over the course of time the safety and efficacy of this procedure and have impressively broadened the scope of this life-saving therapy. But there are many barriers to success. Regimen-related toxicities, graft versus host disease, relapse of underlying malignancy, graft rejection and failure, and of course, the focus of this symposium, barriers to donor availability. And while that is going to remain the focus of this discussion, as the other speakers have nicely outlined, we have to keep these other barriers to success in mind. So it's this crossing the HLA barrier that have really brought us all together today, and how to best do that, getting us to, how can we optimize the use of haplos? Now, we all know, and I'm sure Dr. Parkman and Dr. Reisner kind of summarized this yesterday. You know, the use of haploidentical transplants using ablative or non-ablative conditioning is not new, right? We've been doing this for decades. We've had to overcome, you know, pitfalls to this approach, whether you're keeping T cells in and dealing with graft versus host disease, maybe taking them out in the initial days and dealing with graft rejection, issues with engraftment and infection. And, you know, historically that had led to high failure rates, either from GVHD, relapse, opportunistic infection, et cetera. And that's why it's been so exciting over the last decade or so to see how we could really move this approach forward. So we'll talk about the innovation right here, the use of post-transplant Cytoxan to regulate graft versus host reactions after transplant. So this is a cartoon that many of you may be familiar with. This takes us to day 1 after transplant. And we know that alloreactive T cells are at play here. T cells are becoming activated while they interact with host antigen presenting cells. They then proliferate and make them particularly vulnerable to the approach that involves post-transplant Cytoxan, right? So Cytoxan, being a chemotherapeutic agent, can target these rapidly dividing cells. And we find that these rapidly proliferating alloreactive cells are inactivated. Are they killed? Are they otherwise rendered dysfunctional? That remains to be determined in some ways, but we've made some ground on this. So the hypothesis is that while that is going on with alloreactive T cells, non-alloreactive T cells basically get through this filter and escape post-transplant Cytoxan at day 3 and 4. So in addition, Tregs are part of that non-alloreactive T cell pool, and we've been able to demonstrate in some elegant work published, you know, now almost a decade ago, that these regs increased aldehyde dehydrogenase, and that helps them get through the post-transplant side filter. So these non-proliferating, non-alloreactive cells are spared. They can then lead to infectious immunity, graft versus leukemia effects, and a increase in the population of Tregs. So recently, Waxmuth and colleagues in Chris Kanakari's lab published this very nice paper in the JCI, and they studied the mechanism by which PT-Cy mitigates GVHD in several murine models. What they found is that PT-Cy did not eliminate alloreactive T cells, nor was the thymus required. However, it did induce functional impairment via a variety of suppressive mechanisms that were operative soon after administration. And one of the things they really highlighted was the rapid recovery of alloantigen-specific Tregs. So that kind of, you know, really helped solidify some of the clinical hypotheses. As you know, PT-Cy, at least Hopkins, I don't want to say it was born here, but I think Seattle and Hopkins, you know, were really the two places that pushed this forward. This just kind of summarizes the Hopkins experience, but falls, you know, about a decade short, and we'll highlight some of the things that have gone on since. But in talking to Rick Jones, we've done over 2,000 haploidentical transplants at Hopkins using PT-Cy, and well over 500 in the last 3 years alone. Our data have been replicated now by what I would say are too numerous to count clinical trials and studies that have been published in a variety of wonderful journals, just a small smattering shown here. But interestingly enough, I had a postdoc in my lab do a literature search for post-transplant Cy following allo-BMT, and he actually got 1,700 hits. So certainly a monumental task to try to summarize all of those, but it does speak to the global impact here at 30,000 feet. Our estimates are that approximately 20,000 patients have probably received haplo-BMT with PT-Cy. We're awaiting those final numbers from the CIBMTR, both adult and pediatric patients, and thousands more have now received PT-Cy as a single agent. This graph from the CIBMTR demonstrates the impact of PT-Cy on donor selection. And this dates back from 1980 through 2019. And the inflection point here comes at the end of, or I should say, the end of the 2010 to 2020 period, where some of those initial data were then becoming published, and the phase 2 and phase 3 studies were being done. And that increase in haploidentical donors really kind of was also mirrored by a decrease in the use of cord blood. And this slide from the CIBMTR also shows that PT-Cy is the dominant form of GVHD prophylaxis in the vast majority of these patients undergoing haploidentical transplant, at least in the US. So now that everyone has a donor, what does the future have in store? I think it's exciting, it's bright. So let's talk about some of the data that have been championed by some of my colleagues here, Heather Simons being one of them. You were somewhat familiar with these data. She presented some of them a year or two ago at the pediatric symposium at the at ASBMT. These data were institutional data that were recently published in Blood Advances. This looked at haploidentical transplant using a myeloablative PrEP. The total number of patients in this study were about 100, but about 29 of them were adolescents and young adults. So this is the median age. You see very robust engraftment data. And this outlines what I think has been pretty consistent. Very reasonable and quite low transplant-related mortality, very reasonable and quite low graft versus host disease, particularly acute grade 3 and 4. We're going to see that across a number of studies. Chronic GVHD, particularly moderate to severe, also very reasonable. And here are the initial relapse, overall survival, and event-free survival data. So those data in part were the platform for which this first prospective multi-center trial for pediatric and AYA patients with high-risk leukemia and MDS was launched through the Pediatric Blood and Marrow Transplant Consortium. You guys are familiar with some of these data as well. It was about 32 patients, 60% males. Here's the donor relation, mostly parental, perhaps no surprise. Graft content is shown here, and this range will be important. We'll talk about that in a second. Median follow-up, at least at the time of the slide, is dated as 16 months. And importantly, there were also a number of correlative labs that were being collected from this study to delve into at a later time. So these are some of the data that were generated. Acute graft versus host disease rates here, grade 2, 13%, 3 to 4, none. Moderate to severe chronic GVHD, 11%. When we looked at transplant-related mortality by day 180, this was really impressive. There was none. Relapse is shown here, and this is an issue that has been underscored with a 1 year to 18-month relapse rate of about 30 to 36%. As no surprise, relapse was affected by MRD at the time of transplant, and to some degree also by DRI at the time of transplant. This then shows the overall survival, event-free survival, and graft versus host disease, relapse-free survival here at 1 year and 18 months. You guys are familiar with this. I'm sure this paper is in, this manuscript is in preparation, but these were pretty solid and really demonstrated that this could be done on a multi-center level, as shown here. So the trial really demonstrated safety and feasibility, low accumulative incidences of GVHD, and no transplant-related mortality, limited post-transplant-related complications and viral reactivation. Importantly, there was more graft failure than described in prior studies, and 3 of those patients were untreated patients with MDS. It was important to know that those with graft failure could be salvaged with a second transplant, but again, underscoring that cell dose is important, but more data is certainly needed here. Relapse was something that we want to focus on moving forward, with a trend toward higher relapse rates, both in MRD positive and DRI high patients. And then also demonstrating that it was feasible to collect blood for correlative labs. And this is important because our group, led by Leo Luznik, just recently published a paper that was just now accepted in Blood. Very elegant work using integrative immune profiling and machine-based learning to reveal signatures of graft versus host disease and relapse after post-transplant side. So these data may shed some light on how we may untangle those two issues post-transplant. So what's next from here? Well, we know that relapse is really the final frontier. What have we learned about PT-Cy when used with mycophenolate, mofetil, and tacro? Well, we can shorten the duration of post-transplant immune suppression to as little or as early as day 60 when you're using a haplodonor. And in fact, if you're using it as a single agent in recipients of match-related or unrelated donors, you can essentially be off immune suppression as early as day 5. Now, post-transplant maintenance is perfect here, I think, right? Because you're going to be in a situation where you're off immune suppression, whether we're using cellular therapies that we'll talk about in a second, or medicinal agents. This is going to be particularly promising when combined with reduced intensity transplant conditioning. So this is a trial that is now underway called Excel. This is a phase 2 trial to determine the efficacy of 3 fixed-dose infusions of ex vivo expanded haplo-NK cells. This could be children and AYA patients with high-risk AML undergoing allo-BMT with BuSci and PT-Cy, right? This is so a perfect step off of some of Heather's work. This is going to look at safety, feasibility, efficacy, with a primary objective of relapse-free survival at 1 year. The hypothesis here is that these haplo-NK infusions will improve immune reconstitution and decrease relapse. Chalice Bonifant, who is our institution PI, she is part of the study team, and some of the correlative studies that are going to be done from this trial will be done in her laboratory. Resolve is an interesting protocol we have open here at Hopkins. This is research of expanded multi-antigen specifically oriented lymphocytes for the treatment of very high risk hematopoietic malignancies. Yes, Resolve came out of that. This is a very interesting study that is supported by our program project grant, where we're taking blood from the stem cell transplant donor just prior to the transplant itself. And we're going to generate dendritic cells from this donor blood, and also have peripheral blood mononuclear cells from the donor kind of be stimulated with these antigen pulse dendritic cells. Now, the antigens being used are WT1, PRAME, and survivin. These are antigens that are commonly expressed by AML, but not expressed on healthy tissues. So the goal here is, after an initiation phase, to expand these multi-tumor antigen-specific T cells to be infused after transplant. So that trial is up and running. Our preliminary data have recently been submitted for publication. In addition, COG 2031, this is an ongoing study that's under development, which will be a prospective study comparing MUD versus haplo. I know Heather has really been working hard with the entire group to get this up and running. It'll be a phase 3 randomized trial with myeloablative conditioning. Again, it'll be MUD versus haplo for patients with acute leukemia or myelodysplasia. These patients cannot have a match-related donor. The hypothesis here is that severe GVHD and either 3 to 4 acute or chronic requiring systemic immune suppression will be lower following haplo-BMT, and disease-free survival will not be inferior. There's going to be 2 subsets of patients I'll show you in a second, one randomized to MUD versus haplo and one assigned. And haplo transplant will be really kind of center-committed, center-specific, either using αβT-cell depletion or haplo. So I think this is going to be a great study once it gets kicked off. This is the randomization schema here, where patients who have both a MUD and a haplo donor will be randomized. Arm A will be a haplo-related donor. Again, centers will commit a priori to either T-cell depletion, αβT-cell depletion, or PT-Cy, or they'll be randomized to the MUD arm. Patients who do not have both an 8 out of 8 MUD and a haplo will be assigned to receive a haplo transplant. So really exciting to see this in evolution. So again, how can we move forward? We're always focused on developing a transplant platform that will optimize curative potential for patients in need. We want to overcome all of those traditional barriers. Importantly, this approach should be readily accessible to the majority of transplant centers, not just in the US, but really throughout the globe. I think that transplant using haplo donors and post-transplant Cy has actually addressed several of those traditional barriers. And now the incorporation of reduced intensity regimens really takes it a step forward. Now nearly everyone has a suitable donor, transplant-related mortality in the single digits, and we've talked about the impact on graft versus host disease. But now clearly, this can be completed safely in patients up to 75 years of age. And the vast majority of these data are published in adult recipients. However, this is one of our standard reduced intensity conditioning regimens that we use here at Hopkins. This is a flu-Cy TBI-based regimen. Post-transplant Cy is given on day 3 and 4. And this actually allows this regimen, at least on our adult side, to be given in the outpatient department, and again, routinely used in patients into their 70s. So this was a paper published by previously our Orly Klein, now at Stanford, which really looked at the use of non-myeloablative haplo-BMT in pediatric and young adult patients with hematologic malignancies. It was essentially a proof of principle study. 40 patients that were unable to undergo myeloablative conditioning demonstrated this was feasible with acceptable engraftment, low graft versus host disease, and transplant-related mortality with the overall and event-free survival shown here. How can we continue to build from that? Well, we talked about relapse being the final frontier, but what about using some of these reduced intensity platforms and then coming back with post-transplant maintenance therapy? We have an open trial here in pediatrics and adults using post-transplant blinatumomab for ALL patients, and I'm going to comment here on expanding the scope of haplotransplant to solid tumors, particularly high-risk sarcomas and HPV-positive head and neck cancers. And yes, believe it or not, even prostate cancers in adult males. So Nico Yosa is one of my colleagues who is really committed to trying to determine whether he can use transplant as a platform to awaken a cold and immune-evasive tumor microenvironment to a healthy, unedited donor immune system, thus making that kind of non-inflamed tumors, which many high-risk sarcomas are, more amenable to graft versus tumor effects by cellular infiltration and inflammation. So he initially published some data just a couple of years ago looking at this platform in pediatric patients with solid tumors, again, using a reduced intensity conditioning platform with post-transplant Psi. He demonstrated, again, this was safe and feasible with excellent rates of engraftment, low incidences of non-relapse mortality and GVHD. So this treatment provided a new non-tolerant immune system for post-transplant relapse therapy. So that platform is shown here, a flu-mel TBI-based PrEP. And now at this point, we're stopping immune suppression as early as day 90. We could probably cut that back to day 60. And after a GVHD assessment, these patients are going to be treated with anti-PD-1 therapy. We completed a early part A kind of safety roll-in. We're now accruing patients on part B. This is really the schema, right? Taking patients with dysfunctional T cells, those pre-transplant with tumor growth and relapses due to poor tumor killing, introduce a healthy, unedited donor immune system, mitigate graft versus host disease with post-transplant Cytoxan, and then really focus on graft versus tumor effects, and hopefully through the use of PD-1 blockade, optimize tumor eradication. And some of the correlative data and now the preclinical data that we're doing or generating in the lab is based upon some of these really terrific publications that recently came out of Nico's lab. So have reduced intensity haplodonors and PT-side platforms. Now where can we go? We're interested in expanding the scope of allo-BMT to non-malignant diseases. I'll show you some quick data here in sickle cell disease, beta thal, severe aplastic anemia, also looking at both inherited and acquired disorders of the immune system. For sickle cell disease, this is the platform that we've used. Again, this is a reduced intensity flu-side TBI-based PrEP that incorporates ATG. Our initial publication was in 2012. This was by Dr. Balomas-Mead and colleagues, and basically showed that this is feasible, but graft rejection was still high at 40%. So we come to find that a simple kind of move here by increasing TBI, either the 400 centigrade or keeping it at 200 and adding Thiotepa really made a big difference. So these were data that were recently published in Lancet Hematology. Chris Gamper is one of my colleagues who has championed this effort in pediatrics. This was 17 consecutive patients with either sickle cell anemia or transfusion-dependent thal. 13% were full donor chimeras, 3 were mixed, and only 1 patient had primary graft failure. Rates of acute and chronic GVHD were low. Overall survival, 100%, and transfusion-free survival, 94%. And these Hopkins data, as you know, have gone on to help develop national trials through the BMT-CTN. From a thalassemia perspective, this is just some data that is also a manuscript in preparation. These were initially presented at the ASBMT meeting in 2018 27 patients. This is a busy table. Let me just take you through it quickly. There were 3 basic groups. The first group of patients was 4 patients who received the common regimen of 200, but 2 of those 4 had graft rejection. So we were interested again in augmenting this reduced intensity prep. We looked here at cell dose. We looked at chimerism and count recovery and overall survival in graft versus host disease. I'm going to focus on group A, getting the 400 centigrade of TBI. These numbers are now up to 19 at Hopkins, 16 of which receives haplos. 2 were second transplant secondary to loss of donor chimerism after a prior that occurred at an outside institution. All had great count recovery, 100% overall, and I would say event-free survival. So all of these patients remain transfusion independent. We did not see any VOD, IPS, or TMA, and 2 patients actually had acute grade 2 steroid responsive GVHD. Of interest, 13 of the 16 haplo recipients actually were 100% donor. So let's move on into aplastic anemia. This work has been championed by Amy DeZern, one of my colleagues on the adult side. This was a paper that we published just a couple of years ago together in 2017, looking at primarily haploidentical donors with post-transplant Cy for refractory severe aplastic anemia. These data now were expanded into a publication that I'll highlight in a second, but here's a total of 20 patients with relapsed refractory severe aplastic anemia. Only one of these 20 actually had primary graft failure. This was actually done at an outside institution, but occurred nonetheless. These GVHD rates, again, impressively low and very acceptable. We did have one patient, however, who had severe pulmonary chronic graft versus host disease. Here's some data on CMV and EBV reactivation, really very well tolerated and an acceptable platform here. Amy went on to publish a paper just in Blood Advances a year ago, which expanded on that initial report in BBMT and also included patients who were treatment naive. So this is longer follow-up on those 20 refractory relapse patient and new data on 17 treatment naive patients with severe aplastic anemia. We did see graft failure in 3 of the treatment naive patients, and this was resolved by increasing the TBI from 200 to 400. We used 200 in the relapsed refractory patients, and that worked terrifically well, but perhaps no surprise in these patients who were still immune intact that they required a higher dose of TBI. Here's cumulative incidences of acute and chronic graft versus host disease, again, low and very acceptable, median follow-up of 32 months at the time of the publication with overall and disease-free survival at 94%. And again, increasing that TBI, the naive patients increased engraftment rates terrifically. So these data went on to 2 trials that were run by the BMT-CTN, the relapse refractory trial, many of you may have participated in, that is now complete. And we are about to open a new trial for treatment-naive patients. Moving along as we finish up to patients with non-malignant disorders. Again, this was championed by Orly Klein while she was here. And this was looking at alternative donors with post-transplant Cy for non-malignant disorders. This study that we initially published in 2016, again, a proof of principle that you could use alternative donors in a reduced intensity prep. This was safe and effective for patients with a variety of non-malignant disorders. Of interest, there were about a dozen patients with about 10 different conditioning regimens. So we really wanted to move on. And after the initial 10 or 11, 14 received BMT using this CAMPATH flu-mel-based regimen. And a recent publication of 25 patients with primary immune deficiencies, immune dysregulatory syndromes, or bone marrow failure syndromes was recently published. We're using a more uniform conditioning regimen. Again, prompt neutrophil and platelet recovery, low rates of acute and chronic graft versus host disease. We did have transplant-related mortality in 2 patients, 1 from infection and 1 from VOD after a second transplant. These data were recently published in the Journal of Clinical Immunology, and they now have provided the platform for an open clinical trial using that similar regimen and best available donor, and this has been funded by the Maryland Stem Cell Research Fund. So this was just one step moving forward again at that 30,000 foot level. PT-Cy has been now used for mismatched haplos. These two publications, one in Blood Advances was generated here at Hopkins, and one in the JCO was a CIBMTR trial, both championed by Javier Balones-Mead. PT-Cy is also now being used for peripheral blood stem cell transplants using match-related donors and matched unrelated donors. This was in the form of two clinical trials for the BMT-CTN 1203, the results of which were published in the JCO, where PT-Cy, MMF, and TACRO outcompeted the other two platforms, and that led to BMT-CTN 1703, which I think is about to finish accrual if it hasn't already. PT-Cy has also been used as a single agent for BMT using both match-related and MUDs. This is bone marrow, not peripheral blood. That was a part of BMT-CTN 1301. Those data were recently published. And then just recently published in Blood was a CIBMTR comparison of MUD versus haplo using the PT-Cy MMF TACRO regimen, and really showing that this regimen can be used in several donor sources. So have we changed the standard? Well, I don't think I'm preaching to the converted here. Certainly think we have in the context of using haploidentical donors. I think PT-Cy, in conclusion, what I like to say is elegance is in the simplicity of this approach. Virtually everyone has a donor. This is easy and comfortable to administer. There's no need for sophisticated and costly cell processing or stem cell expansion. This is affordable and transportable worldwide. And I really put that in quotes. I could capitalize it and put it in red. I think that has been the innovation of PT-Cy. And certainly the donor can be mobilized in a timely fashion. And as I've been demonstrating, this is really acceptable and predictable toxicity profiles, not only at Hopkins, but worldwide. So a quote, I believe, by Albert Einstein here: genius is making complex ideas simple, not making simple ideas complex. These are some bonus points that I just touching base. We haven't seen PTLD, no increase in donor-derived malignancies. The number of HLA mismatches really doesn't matter. You can use second-degree haplorelatives, and as I mentioned, unrelated donors that are also haploidentical. We can now safely discontinue calcineurin inhibition by day 60 in patients with malignancies. DLI is also feasible and beneficial. Second transplants can also be used with a different haplo donor and actually results in significant salvage. We have low rates of infection, rapid immune reconstitution. Check out that paper by Dr. Luznick's group that just came out in Blood. Really some elegant work there. Just as I mentioned, PT-Cy has really been used as a platform for several national haplo initiatives, some of which are commented on here. Really excited about that PBMTC trial and the upcoming COG trial. So this just gives you an idea of the development of PT-Cy over the years at a couple of centers, initially here in the US, and how really over the last decade or so, this has just taken off, where there are multiple centers throughout the globe now using PT-Cy as the preferred platform for haploidentical transplants. So with that, on my summary slide here, we know that allogeneic transplant is a curative option. Innovations over time have reduced regimen-related toxicity and really shattered issues related to donor availability. PT-Cy certainly allows the use of T-cell replete bone marrow from haploidentical donors, can also be used as a single agent. Thousands of patients now receiving this platform nationwide. We think it's an optimal platform for post-transplant maintenance therapies and really opportunities to extend transplant to a more and more patients in need across the globe. And again, I wanted to highlight how institutional data driven by many of my colleagues here at Hopkins has really led to the platform for several multi-center trials. So in concluding, and just an acknowledgement, no question it takes a village. This is a little dated here, but this is our BMT team here at Hopkins. Also want to make sure I acknowledge my colleagues in both the adult BMT program and our clinical trials office, and no doubt to our patients and their families who we are all committed to treating and owe a debt of gratitude. So thank you very much for your attention, and thank you again for inviting me to be a part of this wonderful symposium.
Thank you, Ken. That was great. We'll now start entertaining questions. So any of you who have questions, go ahead and put them in the chat. Ken, the first thing I want to say is happy birthday. Thank you for sharing your birthday with us. We appreciate that.
You shouldn't have told everyone that we were residents and fellows together. Now I can't hide my age. You know, it's you would blow my cover.
You got it. All right. So I want to start off with a little bit of a provocative question. Franco and Brandon, Ken has just told you that post-transplant cyclophosphamide has made a global impact, which it has. And, you know, he's kind of laid down the gauntlet here, saying this is simple, it's elegant, and yet express dissatisfaction. I see that obviously, Ken. It's not just post-transplant Cy, you need to do something to prevent relapse afterwards. But why should we then do more sophisticated, either alpha beta or CD45RA depletion that could be potentially more expensive? I want to hear from the two of you what you think are really selling points as to why we should be doing more sophisticated stem cell manipulation. Start with Franco. You're first, Franco.
Thank you so much, Mike, for raising the question. Of course, the post-transplant cycle is very simple and potentially suitable to be adopted across many countries and worldwide, while you need a certain level of qualification and expertise in the graft manipulation. But I don't think that the question can simply be addressed in this way. You have also to consider the post-transplant complications that the patients have, the length of hospitalization, the supportive care that the patients need. And at the end of the day, I am not so convinced that the balance is in favor of the post-transplant cyclophosphamide. In the few cases that we perform, there were significant, in many patients with lymphoma, there were significant burden of complication, including hemorrhagic cystitis, rather than very prolonged hematopoietic recovery that render the approach a little bit more complicated. Our median hospital length stay is in the order of 20-25 days, despite the fact that we use a myeloablative conditioning regimen. And again, to be provocative, I don't think that a non-myeloablative regimen can have a great room in the management of children with acute leukemia. I fully admit that the approach can be much more attractive for adult patients in whom the risk of complication associated with a very intensive and aggressive conditioning regimen is certainly greater than that we see in the pediatric population. But at the end of the day, I think that if you are able to implement a good graft processing approach, the αβT-cell depleted platform can offer several advantages, and in part of them are also shared through the approach of CD45RA depletion. Thank you.
Thank you. Brandon, what do you have to say?
Well, I'll start by saying that we do post-transplant Cy here as well. So, you know, it's a very powerful way to create tolerance, and we utilize it, particularly in our non-malignant population. I've also used it for mismatched MUDs when they have no haplo options and no matched donors. We've used it a few times. So, you know, I think it has a great place, and it really facilitates both engraftment and low graft versus host disease. I'm also intrigued by, Ira Reisner, and I think Rupert's done a few patients with both T-cell depleted graft and post-Cy. And can we have a super tolerance inducing regimen? That would be interesting in some situations like solid organ transplant tolerance induction and stuff like that. So there's definitely a place for it, and we've done it. So we've also done it in our high-risk patients that we get here at our institution. I wrote a protocol a few years ago for patients getting second transplant and really tried several mechanisms to intensify the regimen and try to intensify the post-transplant GVL with using post-Cy. It did not work, at least the way we tried. The publication, I wasn't super thrilled about publishing it because the outcomes aren't great, but it will be published hopefully soon. And so I think relapse is a problem. And then I think the real advantage to me of doing graft manipulation is you can set the type of cells, in theory, the right types of populations, the right proportions of the different immune populations, and the right doses at the right time in the early post-transplant period. And so, post-transplant Cy kind of settles the immune system down, and then you build from there. And maybe you can, as Kenneth mentioned, which is really intriguing, add additional cell therapies after that. Of course, you don't really want to be on calcineurin inhibitors for a long period of time. But even then, I'm still using GVH prophylaxis in some of my studies for a short period of time. And so can we get to a place where we can choose the right types of populations, get them early when the GVL effect should be happening? It has to happen early in these aggressive malignancies. The average time to relapse in refractory patients and second transplant patients is by 4 months. So we have to get GVL as soon as possible. We have to be protected from infection as soon as possible. We have to do it in a way that minimizes or modulates graft-versus-host disease risk. So it's definitely has a place, and as you saw, it's a huge benefit for the whole world, and so I'm happy to have it. The one thing I would add is that I have had international physicians contact me and say they can't do post-Cy locally when they ask for advice about a patient, and I suggest it. Cytoxin is available most places. Some places don't have it. Calcineurin inhibitor, ability to do levels, things like that. So they actually, some places are a little bit easier to do. If they can get the device and the reagents, then they do alpha-beta depletion, and they don't need post-transplant immune suppression. That's easier for some places. And so there's certainly, for low and middle income countries, I think we have a lot of barriers to being able to do transplant like we do here in high income countries.
Yeah, that makes sense. Let me give a couple more questions from our audience here. First is from Tatiana Saldarriaga. I'm going to mess up your name, sorry. Is there any clinical trial ahead, Cy, post-transplant Cy versus alpha-beta CD19 BMT depleted cells in adults or children? Now, I'll actually jump in and answer. Ken pointed out the trial that Heather Simons and actually Alicia is also chairing and co-chairing through the Children's Oncology Group. That study is not designed to compare post-transplant Cy with alpha-beta. However, it will have 2 very large cohorts of alpha-beta and post-transplant Cy done at the centers that have committed to each of those approaches. And that may give us a head-to-head comparison. I would love to have more trials actually compare them head-to-head. Ken, here's a direct question to you from Carmen. Ken and all, excellent lectures. Thank you all, dear friends. Dr. Cooke, can you elaborate on why we have so much more CNV reactivation and BK hemorrhagic cystitis after the haplos using post-transplant Cy? Haplo post-transplant Cy has changed the outcome of patients transplanted in Brazil and other countries with limited resources. It was a turning point for our patients. Thank you, Carmen. But Ken, what do you say about the CNV reactivation and the BK hemorrhagic cystitis?
These are great questions, and let me just say regarding the other platforms, we haven't gotten here without people really pushing the borders and doing really terrific work to continue to see how we can improve on our approaches for transplant for patients. So I applaud everyone's effort in doing that, and I just wanted to kind of set perhaps set PT size as the standard or the benchmark to where we can compare some of these things. But this is one of the important issues. What about opportunistic infection, viral reactivation? And some of the data that we had just shown, whether it be in malignant or non-malignant disorders, those incidents of EBV and CMV reactivation were actually relatively low and acceptable. So it's a really good question. We generally don't have a big problem with CMV reactivation. We see it in the context of those patients who are at risk. We, although in the big picture, not matching for CMV donor and host doesn't really have a big impact on outcomes. I think this has been published. We still try, if we have a CMV positive patient, we're still looking for a CMV positive donor, at least our group is, for this reason, right? If we're going to see viral reactivation, we want to be able to control it. You know, whether you want to suggest that CMV can help with some proliferation of alloreactive cells in a good way, I would rather not be dealing with that. So I don't have a good answer for Carmen in that regard. Obviously, we have super high vigilance for following and tracking these patients post-transplant to look for viral reactivation. We have a very sensitive test, so we generally wait until we're going to see repeated and increasing levels before we even pull the trigger. And I will tell you, my adult colleagues will not treat CMV reactivation in the pre-engraftment period because their adult patients generally don't tolerate foscarnet very well, and they don't like giving ganciclovir. So they've recently published these data that you can get by without treating CMV reactivation, but no question that raises the hair on the back of our neck. For hemorrhagic cystitis, it's another issue. Is this related to BK reactivation? Is it a product of post-transplant Cytoxan? It is a good question. We see it, we look for it. It has not been a huge barrier to our outcomes, but maybe we can put our heads together offline and see how we can address some of those issues.
Thank you, Ken. Now, I wanted to ask one quick question. There are a number of questions that have poured in, and hopefully the panelists can please try to answer them in the chat if we don't get to them. But Franco, you said something that really intrigued me and showed data to back it up, and that is that if patients have had active or recent infections and they're going into this type of a procedure, they do poorly. Now, that should be obvious. But what does recent mean? I want to know how long you want someone to be infection-free before you try to do this procedure. I'm just really curious, and I'd love to hear that from all of you. How infection-free do you need them to be to undergo a haplotransplant?
So in the analysis that I presented, we evaluated the occurrence of infection either at time of the allograft or in the 3 weeks before the transplant procedure. So the poorer outcome were observed in those children who had either an active infection or an infection occurring in this time interval, if you wish. And the other point that I'd like to briefly mention, considering that I have the opportunity to further elaborate a little bit, is that with the αβT-cell depleted approach, the incidence of GVHD is amazingly low. And you probably noticed that the GVHD relapse-free survival was more or less the same of the disease-free survival. And that's why I think that we should be very accurate in estimating the relative cost of the procedure. And I fully agree with you, with Ken and Brandon, that we need prospective trials to have a reliable answer on the relative pros and cons of the different platforms.
Great. I want to just ask 30 seconds because I want to finish this question. Ken, Brandon, if you have someone who has an active infection, do you take them to transplant? If you if they had a recent infection, how long do you make sure they're infection free before you take them to A haplo and you know they're going to have bad immunity for at least 6 to 9 months? Just a quick 20-second answer. Brandon first.
So bacterial infection, we want them cleared of that because it can cause a lot of post-transplant inflammation. So I think we're mostly talking about viruses. We worry greatly about viruses. I would say it depends on the virus. If we feel like they can clear it in the next couple of weeks, we try to let them clear it, but sometimes there's a risk benefit. We have taken patients with active, so on our haplo trials that give memory cells, we've taken a patient that had active EBV PTLD. It wasn't really PTLD because they never had a transplant, but it was EBV driven lymphoma. So we were very worried about overproliferation. We took a patient with splenomegaly, hepatomegaly, and active CMV that we could not clear, and was very worried about the memory cells kicking in too strongly. They both cleared their infections and their refractory leukemia. So those are the success stories, but man, we were very nervous. We've had patients with HHV6 and the memory cells kick in and they have very severe liver inflammation, hepatitis and stuff like that. And so, ideally you want as low of antigen burden as possible, whether that be leukemia antigen or viral antigen, so that the cells come in and do more like that patient example I gave you. They kind of clear the virus, the low amount of virus without us ever knowing it. But sometimes your back's against the wall and you got to do what you got to do.
Absolutely. Ken, last word on your birthday.
Yeah, so I think it's a great question. I think it really depends on a couple of things. Underlying diagnosis, right? I mean, if someone's got beta thalassemia and there's not an urgency, we're going to make sure that patient is as in good shape as possible. However, if you have immune dysregulatory or immune deficiency, aplastic anemia, the only way you're going to get over those infections is to have a healthy immune system. And sometimes we just got to go through. The second thing is, what is the platform? Are we using serotherapy or not? If we're using ATG or Campath, and boy, we're going to really try to work hard to get those patients as clean as possible. And, we have our surveillance up as part of our pre-transplant workup, where we're looking at these patients under a microscope to try to identify any evidence for occult infection going in, and we try to stamp it out as quickly as possible. So I would say if we can, a couple of weeks, 2 to 3 weeks, you'd like to know these patients are going in the transplant. Of course, COVID comes up always at the very last minute, but we want to try to get them as infection-free as possible if we're able.
Absolutely. Well, excellent responses. I wish we had another half hour to ask questions, but we're past our time. I'd like to again invite the people on the panel to please respond to some of the questions that are still in the Q&A in writing, if you would. Yes.
Mike, since we are going to the break, if the speakers are okay, maybe we can still answer just a couple of questions in the Q&A. I think that the audience is really super excited. So if you are okay, I would just take a couple of questions that you can pick.
All right, we'll do it really quickly then. Here's an interesting one from Anant, but cyan, is post-transplant Cy really cheaper than alpha T-cell depleted in the long run? If post-transplant Cy has a higher risk of viral reactivations. And first, I'm not sure if post-transplant Cy has a higher risk of viral reactivations. It may or may not. But what's cheaper, alpha beta versus post-transplant Cy?
Well, I would agree with you, Mike, on your first comment. I'm not sure there's any data to necessarily say that there is a higher incidence of opportunistic infection or viral reactivation. But, it's really a balance. Franco mentioned, you have to keep in mind how long are these patients going to be in the hospital. Well, I will tell you, our adults, and we are pediatricians, but we can't just think about kids. Our adults, this is done in the outpatient clinic. So if you want to talk about really reducing costs, those patients aren't admitted at all. And I think we should consider getting our pediatric patients to reduced intensity conditioning regimens, maybe not ALL right out of the gates, but certainly our non-malignant disorders, our aplastic anemics, et cetera. They do not have prolonged hospital stays. So you got to balance everything that's going on post-transplant. And I would just say, listen, if you don't have the money to get the patient an alpha beta T-cell depletion from the get-go, and there's now published reports that there are certain countries that will look at the cost and that will be as expensive as an unrelated donor. They just can't do it. So then transplant is not even an option. So I think you have to weigh the costs on the front end, on the back end. And our job is to make this as safe and effective as possible so we don't have patients in the hospital for weeks and weeks.
Can I just
So do you think it's cheaper? Oh, throw it in, Brandon. Yep.
Well, I was just going to say, I mean, volume matters, right? And so if we figure out the absolute best way to do transplant and it works beautifully and everyone in the world adopts it, it's going to get cheaper. Whatever it is going to get cheaper due to volume. So if it's graft manipulation, someone's going to invent a cheaper way to do the graft manipulation that works. We have to figure out the best way to do it, and it'll get cheaper.
Well, one comment to make from my own studies on this, and we have a financial analysis that we're doing, but it's going to take a while before we get it out there. And unfortunately, it's not compared directly with post-transplant Cy, but you know, patients have no immune suppression post-transplant with the alpha beta that we do, and they go home very early. So you know, 2 days of hospitalization makes up for the $15,000 to $20,000 that we have to pay for the graft manipulation.
Is that
any
a secondary
comments, Franco?
aim, or could it be in the COG trial, Mike and Alice?
It should be. We've talked about it. Alice, I don't know if we formally added it. Do you remember?
Yes, we included in the prospective COG trial. Yes, so we will
Excellent.
have this information collected as well.
Nothing like data
Yeah,
to
well,
put things to rest, right?
Yeah.
Exactly right. Here's one other question that I want to ask that's incredibly important. What has been the experience of each of you with post-transplant cytopenias? Because this is definitely an issue with this type of transplantation. So I don't know who wants to go first. Franco, let's let you start. Autoimmune cytopenias. Franco,
in the alpha beta setting, the post-transplant cytopenia is a relatively rare event. There are few cases mainly occurring in patients with mixed chimerism of autoimmune hemolytic anemia that we have observed, but usually this type of complication doesn't occur in the setting of alpha beta T-cell depleted transplant. Let me be a little bit more provocative again for defending the alpha beta platform. There is a still unpublished analysis of the inverse error working party of the αβT, in which Maris Latter and a colleague of mine, Dr. Algeri, collected data of children with PIDs treated either with the alpha beta or with the post-transplant cyclophosphamide across the different European countries. And there is an advantage in the probability of disease-free survival statistically significant in favor of patients given an alpha beta T-cell depleted transplant.
Okay. Brandon, what's your comment on cytopenias, autoimmune cytopenias?
Yeah, I think we might have seen one. We see the occasional autoimmune cytopenia 1 or 2 every year or two, and they don't seem to be more frequent in our household than our other transplants.
Interesting, not a problem in your mind. Ken, what's your thought?
Yeah, we've seen it also relatively infrequently. The interesting thing is that this was a paper published in BBMT just a couple of years ago. We've seen that if you do flow, peripheral flow, that you can sometimes see a large granular lymphocytosis in these patients. And that's very amenable to a short pulse of steroids. So that paper was also published from my colleagues on the adult side. So yes, we see it pretty rarely. And that's the first thing we would actually look to see if we have any evidence of LGL. And if so, we may treat that, again, with a brief pulse and taper of steroids, generally successfully.
Yeah, so this is interesting, but I think an important thing that this has brought up is that we need to look at these type of complications and really document whether it is more common. In my experience, it's not more common after alpha beta or post-transplant Cy. But it'd be nice to, as Ken said, it'd be nice to have a little data to back us up.
I have seen a little, I'm sorry, I just want to comment because I have seen a little bit of delayed hemolytic reaction and minor ABO mismatch in our post-side transplants. And I would get Ken's thought on that. Because we're giving all these lymphocytes, and when we see minor ABO, the first patient really kind of surprised us, and I didn't know if the questions had to do with that. So, I mean, that's a self-limited thing. Once all of the previously transfused pre-transplant red cells go away and you switch to full donor, the hemolysis goes away, but it can be pretty. Our first patient we were unprepared for was pretty impressive hemolysis.
Was that a malignant or non-malignant and myeloablative or reduced intensity prep?
It was very intense, but submyeloablative technically, reduced intensity with PBSC graft and only 1 dose of post-cy.
Yeah, we do try to avoid in our sickle cell patients in particular who are getting a reduced intensity prep and maybe allo-immunized. We do try to really avoid major mismatches in ABO in that context. That's really the place that we've seen it a little bit, and we've tried to kind of get away from that. And of course, using haploidentical donors, we generally have more choices, right? Which is great, regardless of the platform we're using for GVHD prevention.
Yeah, I think that's an important point. We too also totally avoid ABO mismatch if we can, if possible. Well, again, I think those are the key questions that we wanted to go over. So outstanding session. I look forward to more data, prospective trials, and also to the wonderful advanced trials, really exploring these unique therapies that each of you and many of us who are listening to these talks are doing. So thank you so much for this great session.
Thank you, really a pleasure. Thanks for the opportunity.
Thank you. Thank you so much. All my best.
Thanks, everyone. I'll see you all back at 10 a.m.